Institute for Advanced Studies, Jodhpur 3. Faster than when either of wavelengths alone was provided, this synergism or enhancement became known as the Emerson enhancement effect. These two observations Institute for Advanced Studies, Jodhpur 3. Faster than when either of wavelengths alone was provided, this synergism or enhancement became known as the Emerson enhancement effect. Observations: the red drop effect and enhancement effect led to the first indication that the light reaction has two sites of action, one in the red region of the spectrum and the other in the far. The explanation offered for these two effects is that the light reaction actually consists of two photoacts, photoact I and photoact 2, mediated by two photosystems, photosystem I and photosystem 2. Photosystem I is driven by the far red light, and when it operates alone, it produces the red drop. But when it operates along with photosystem 2, which functions in the red region, enhancement effect is produced. The function of the two photosystems had been successfully carried out and their functions clarified. Photosystem I has been located in the thylakoid membranes. It is made up of three forms of light, one absorbing maximally at 683 nanometers, the second absorbing maximally at 695 nanometers, and the third at 670 nanometers. The last of these has been called P100. Photon had been located in the stroma thylakoids. It is made up of two forms of chlorophyll with maximum absorption at 670 and 690 nanometers. The second form is Crescent P690. Each photosystem I a reaction center made up of a special chlorophyll molecule. In photosystem I it is a protein-bound chlorophyll molecule, P700. In photosystem 2 it is P690. The reaction centers are the actual sites where light energy is converted to chemical energy. Electron carriers in photosystem I, X, plastocyanin, cytochrome F and ferredoxin is the electron carrier. Photosystem 2 has plastoquinone and cytochrome B500P9. E other chlorophyll and carotenoids, which merely serve to transfer the light absorbed by them to the active centers. Photosystem takes in both cyclic and non-cyclic photophosphorylations. PSI can carry on cyclic photosphorylation independently. Normally it drives an electron from photosystem 2 to NADP+. Photosystem 2 picks up electron release during photolysis of water. The same is extruded on absorption of light energy. The extruded electron passes over cytochrome complex. Sufficient energy is released to take part in the synthesis of ATP from ADP and inorganic phosphate. This photophosphorylation is non-cyclic. PS2 can operate only in conjunction with PSI. Phase 2, energy transduction. The excited molecules of P700 and P690 transduce their energies to generate ATP and NADPH2. Molecular oxygen is also produced, but it escapes out of the photosynthetic system. AT and NADPH2 together constitute the assimilatory power and are employed in the fixation of CO2 in the dark reaction. Photophosphorylation. Photophosphorylation is the light-driven or light-energized synthesis of ATP. It was discovered by Amin et al. In 1954. Photophosphorylation is of two main types, cyclic and non-cyclic. Photophosphorylation. It is a process of photophosphorylation in which an electron expelled by the excited photocenter is returned to it after passing through a series of electron carriers. Cyclic photophosphorylation is performed by photosystem I only. Its photocenter P700 extrudes an electron with a gain of 23 kilocalories per mole of energy after absorbing a photon of light, HV. After losing the electron, the photocenter becomes oxidized. The expelled electron through a series of carriers including X, ferredoxin, plastoquinone, cytochrome complex and plastocyanin before returning to photocenter. While passing between ferredoxin and plastoquinone and, or over the cytochrome complex, the electron loses sufficient energy to form ATP from ADP and inorganic phosphate. Institute for Advanced Studies, Jodhpur 4. Halobacteria or halophile bacteria also perform photophosphorylation, but ATP thus produced is not used in synthesis of food. These bacteria possess purple pigment bacteria or rhodopsin attached to plasma membrane. Light falls on the pigment, it creates a proton pump which is used in ATP synthesis. Non-cyclic photophosphorylation. It is the normal process of photophosphorylation in which the electron expelled by the excited photocenter does not return to it. Non-cyclic photophosphorylation is carried out in collaboration of both photosystems I and II. Electron released during photolysis of water is picked up by photocenter of PS2 called P680. The same is extruded out when the photocenter absorbs light energy, HV. The extruded electron has an energy equivalent to 23 kilocalories per mole. It passes through a series of electron carriers Q, PQ, cytochrome complex and plastocyanin. While passing over cytochrome complex, the electron loses sufficient energy for the synthesis of ATP. The electron is handed over to photocenter P700 of PSI by plastocyanin. P700 extrudes the electron after absorbing light energy. The electron passes through X, Phase center A, ferredoxin, and NADP reductase, which combines it with NADP+ for becoming reduced H+ released during photolysis to form NADPH. ATP synthesis is not direct. The energy released by electron is actually used for pumping H+ ions across the thylakoid membrane. It creates a proton gradient. The gradient triggers the coupling factor to signal 1 E-size ATP from ADP and inorganic phosphate. Oxygen evolution. The oxygen that is evolved during photosynthesis comes from water and it is part of the photoact 2 mediated by PSI. The light energy trapped by this system excites P690 and 2 electrons are ejected. The energy is used to remove 2 electrons from the hydrogen of the water and boost them to a higher energy level. At this point, the molecular oxygen which escapes from the photosynthetic system is formed. It through the carriers plastoquinone PQ, cytochrome B559, cytochrome F, plastocyanin and finally end up in P700 to take it back to the ground state. In photosystem 2, the electrons that brings the excited chlorophyll molecule to the ground state comes from photolysis of water. The aspect of oxygen evolution during photosynthesis is its relationship to the presence of certain ions in the medium such as Cl, Mn2+ and bicarbonate. 2H2O2 plus 4H plus 4E. Both the photons working in union produce, for every 2 turns, 2 molecules of NADPH2, 3 ATPs, and a molecule of oxygen from 2 water molecules. Some herbicides block electron flow. The use of herbicides to kill unwanted plants is widespread in modern agriculture. Many different classes of herbicides have been developed, and they act by blocking amino acid, carotenoid, or lipid biosynthesis or by disrupting cell division. Other herbicides, such as DCMU, dichlorophenyldimethylurea, and paraquat, block photosynthetic electron figure. DCMU is also known as styrene. Paraquat has acquired public notoriety because of its use on marijuana crops. Herbicides, DCMU among them, act by blocking electron flow at the quinone acceptors of photosystem II, by competing for the binding site of plastoquinone that is normally occupied by QB. Such as paraquat, act by accepting electrons from the early acceptors of photosystem I and then reacting with oxygen to form superoxide, O2, a species that is very damaging to chloroplast components, especially lipids. Carotenoids serve as photoprotective agents. In addition to their role as accessory pigments, carotenoids play an essential role in photoprotection. The photosynthetic membrane can easily be damaged by the large amounts of energy absorbed by the pigments if this energy cannot be stored by photochemistry. This is why a protection mechanism is needed. The photoprotection mechanism can be thought of as a safety valve, venting excess energy before it can damage the organism. When the chlorophylls in the excited state is rapidly dissipated by excitation transfer or photochemistry, the excited state is said to be quenched. Institute for Advanced Studies, Jodhpur 5. Institute for Advanced Studies, Jodhpur 6. The state of chlorophyll is not rapidly quenched by excitation transfer or photochemistry. It can react with molecular oxygen to form an excited state of oxygen known as singlet oxygen, 1O2*. The extract of singlet oxygen goes on to react with and damage many cellular components, especially lipids. Carotenoids exert their photoprotective action by rapidly quenching the excited state of chlorophyll. The excited carotenoids does not have sufficient energy to form singlet oxygen, so it decays back to its ground state while losing its energy as heat. Mutant organisms that lack carotenoids cannot live in the presence of both light and molecular oxygen, a rather difficult situation for an O2 evolving photosynthetic organism. For an O2 evolving photosynthetic bacteria, mutants that lack carotenoids can be maintained under laboratory conditions if oxygen is excluded from the growth medium. Recently, carotenoids were found to play a role in non-photochemical quenching, which is a second protective and regulatory mechanism. Some xanthophylls also participate in energy dissipation. Non-photo quenching, a process regulating the delivery of excitation energy to the reaction center, can be thought of as a volume knob that adjusts the flow of excitations to the PSII reaction center to a manageable level, depending on the light intensity and other conditions. The process appears to be an essential part of the regulation of antenna systems in most algae and plants. Non-photochemical quenching is the quenching of chlorophyll fluorescence by processes other than photochemistry. As a result of non-photochemical quenching, a large fraction of the excitations in the antenna system caused by intense illumination are quenched by conversion into heat. Non-photochemical quenching is thought to be involved in protecting the photosynthetic machinery against overexcitation and subsequent damage. The molecular mechanism of non-photochemical quenching is not well understood, although it is clear that the pH of the thylakoid lumen and the state of aggregation of the antenna complexes are important factors. Carotenoids, called xanthophylls, are involved in non-photochemical quenching, violaxanthin, anthraxanthin, and zeaxanthin. In high light, violaxanthin is converted into zeaxanthin, via the intermediate anthraxanthin, by the enzyme violaxanthin depoxidase. When light intensity decreases, the process is reversed. Binding of zeaxanthin to light harvesting antenna proteins is thought to cause conformational changes that lead to quenching and heat dissipation. Non-photochemical quenching appears to be preferentially associated with a peripheral antenna complex of photosystem II, the PSBS protein. The photosystem II reaction center is easily damaged. Another effect that appears to be a factor in the stability of the photosynthetic apparatus is photoinhibition, which occurs when excess excitation arriving at the PSII reaction center leads to its inactivation damage. Photoinhibition is a complex set of molecular processes, defined as the inhibition of photosynthesis by excess light. Photoinhibition is reversible in early stages. Prolonged inhibition, however, results in damage to the system such that the PSII reaction center must be disassembled and repaired. The main target of this damage is the D1 protein that makes up part of the PSII reaction center complex, see figure. When damaged by excess light, it must be removed from the membrane and replaced with a newly synthesized molecule. The PSII reaction center are not damaged by excess excitation and are thought to be recycled, so the D1 protein is the only component that needs to be synthesized. Photosystem I is protected from active oxygen species. Photosystem I is particularly vulnerable to damage from active oxygen species. The ferredoxin mixture of PSI is a very strong reductant that can easily reduce molecular oxygen to form superoxide, O2. This reduction competes with the normal channeling of electrons to the reduction of NADP plus and other processes. Superoxide is one of a series of active oxygen species that can be very damaging to biological membranes. Superoxide formed in this way can be eliminated by the action of a series of enzymes, including superoxide dismutase and ascorbate peroxidase. Thylakoid stacking permits energy partitioning between the photosystems. Photosynthesis in higher plants is driven by two photosystems with different light-absorbing properties poses a special problem. The flow of energy to PSI and PSII is not precisely matched and conditions are such that the rate of photosynthesis is limited by the available light. Low light intensity, the rate of electron flow will be limited by the photosystem that is receiving less energy. In the most efficient situation, the input of energy would be the same to both photosystems. However, no single arrangement of mints would satisfy this requirement. Institute for Advanced Studies, Jodhpur 7. Because at different times of day the light intensity and spectral distribution tend to favor one photosystem or the other. This problem can be solved by a mechanism that shifts energy from one photosystem to the other in response to different conditions. Such a regulating mechanism has been shown to operate in different experimental conditions. The observation of quantum yield of photosynthesis is nearly independent of wavelength strongly suggests that such a mechanism exists. Thylakoid, a protein kinase that can phosphorylate a specific threonine residue on the surface of LHCII, one of the membrane-bound antenna pigment proteins described earlier in the chapter. LHCII is not phosphorylated, it delivers more energy to photosystem II, and when it is phosphorylated, it delivers more energy to photosystem I. Kinase is activated when plastoquinone, one of the electron carriers between PSI and PSII, accumulates in the reduced state. Reduced plastoquinone accumulates when PSII is being activated more frequently than PSI. The phospholipid II then migrates out of the stacked regions of the membrane into the unstacked regions, probably because of repulsive interactions with negative charges on adjacent membranes. The migration of LHCII shifts the energy balance toward photosystem I, which is located in the stromal lamellae, and away from photosystem II, which is located in the stacked membranes of the granule. This situation is called state II. More oxidized because of excess excitation of photosystem I. The kinase is deactivated and the level of phosphorylation of LHCII is decreased by the action of a membrane-bound phosphatase. LHCII then moves back to the grana, and the system is in state I. This is a very precise control of the energy distribution between the photosystems, allowing the most efficient use of the available energy. Blackman's reaction. Phase 3, energy stabilization. Of carbon dioxide to a carbohydrate is the essence of this phase, and this is accomplished through the employment of the assimilatory power ATP and NADPH2 generated in the light reaction. This energy stabilization is a dark reaction. Light. Instead, assimilatory power ATP and NADPH2 produced during photochemical phase is used here in fixation and reduction of CO2. The enzymes required for the process are present in the matrix or stroma of the chloroplast. There are two MAM pathways for the biosynthetic or dark phase Calvin cycle and C4-dicarboxylic acid cycle. The plants exhibiting the two are respectively called C3 and C4 plants. Calvin cycle, the photosynthetic carbon reduction cycle, or the C3 photosynthetic pathway. It was discovered by Calvin, Benson and their colleagues using unicellular algae chlorella and C-Nesmus obliques and radioactive isotope of 14C with a half-life of more than 5,000 years. Phases of Calvin cycle. Calvin cycle is divided into the following three phases: carboxylation, glycolytic reversal and regeneration of RUBP. 1. Carbon requires ribulose 1,5-biphosphate or RUBP is acceptor of carbon dioxide and RUBP carboxylase or Rubisco's enzyme. The enzyme was previously called carboxydismutase. Carbon dioxide combines with ribulose 1,5-biphosphate to produce a transient intermediate compound called 2-carboxy-3-keto-1,5-biphosphoritol. The intermediate splits up immediately in the presence of water to form the two molecules of 3-phosphoglyceric acid or PGA. It is the first stable product of photosynthesis. 2. Glycolytic reversal. The processes involved in this step or phase are reversal of processes found during glycolysis, part of respiration. Phosphoric acid or PGA is further phosphorylated by ATP with the help of enzyme triphosphate kinase. It gives rise to 1,3-phosphoglyceric acid. 2-phosphoglyceric acid is reduced by NADPH through the agency of enzyme triphosphate dehydrogenase. It produces glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde. Glyceraldehyde 3-phosphate is a key product which is used in synthesis of both carbohydrates and fats. Performing carbohydrates, say glucose, a part of it is changed into its isomer called dihydroxyacetone 3-phosphate. The enzyme that catalyzes the reaction is phosphate isomerase. The two isomers condense in the presence of enzyme aldolase forming fructose 1,6-diphosphate. 6-diphosphate POP loses 1-phosphate group, forms fructose 6-phosphate F6P, which is then changed to glucose 6-phosphate G6P. The latter can produce glucose or become part of sucrose and polysaccharide. As glucose is a 6-carbon compound, 6 turns of Calvin cycle are required to synthesize its one molecule. 3. Regenerate fructose 6-phosphate F6P and glyceraldehyde 3-phosphate gap react to form erythrose 4-phosphate E4P and xylulose 5-phosphate X5P. Erythrose 4-phosphate combines with dihydroxyacetone 3-phosphate to produce acetoheptulose 1,7-diphosphate STP, which loses a molecule of phosphate in. Institute for Advanced Studies, Jodhpur 8. Gives rise to acetoheptulose 7-phosphate S7P. Acetoheptulose 7-phosphate reacts with glyceraldehyde 3-phosphate to produce xylulose 5-phosphate X5P and ribose 5-phosphate R5P. Both of these are changed to their isomer ribulose 5-phosphate Ru5P. Ribulose 5-phosphate picks up a second phosphate from ATP to become changed into ribulose 1,5-biphosphate RUBP. Institute for Advanced Studies, Jodhpur 9. Regulation of the Calvin cycle. The high energy of the Calvin cycle indicates that some form of regulation ensures that all intermediates in the cycle are present at adequate concentrations and that the cycle is turned off when it is not needed in the dark. In general, variation in the concentration or in the specific activity of enzymes modulates catalytic rates, thereby adjusting the level of metabolites in the cycle. Changes in gene expression and protein biosynthesis regulate enzyme concentration. Post-translational modification of proteins contributes to the regulation of enzyme activity. Genetic level, the amount of each enzyme present in the chloroplast stroma is regulated by mechanisms that control expression of the nuclear and chloroplast genomes. Term regulation of the Calvin cycle is achieved by several mechanisms that optimize the concentration of intermediates. These mechanisms minimize reactions operating in opposing directions, which would waste resources. Two general mechanisms can change the kinetic properties of enzymes. 1. Covalent bonds such as the reduction of disulfides and the carbamylation of amino groups, which generate a chemically modified enzyme. 2. Benevolent interactions, such as the binding of metabolites or changes in the composition of the cellular milieu, e.g., pH. In addition, enzymes to the thylakoid membranes enhances the efficiency of the Calvin cycle, thereby achieving a higher level of organization that favors the channeling and protection of substrates. Calvin cycle is not a dark reaction. Light-dependent enzyme activation regulates the Calvin cycle. 5 light-regulated enzymes operate in the Calvin cycle. 1. Rubisco. AP, glyceraldehyde 3-phosphate dehydrogenase. 3. Fructose 1, 6-bisphosphatase. 4. Dehydrolose 1, 7-bisphosphatase. Ribulose 5-phosphate kinase. The last 4 enzymes contain one or more disulfide S groups. It controls the activity of these 4 enzymes via the ferredoxin-thioredoxin system, the covalent thiol-based oxidation reduction mechanism identified by Bob Buchanan and colleagues. In the dark, these residues exist in the oxidized state S, which renders the enzyme inactive or subactive. In the light, the S group was reduced to the sulfhydryl state HHS. This redox change leads to activation of the enzyme. Of the crystal structure of each member of the ferredoxin-thioredoxin system and of the target enzymes fructose 1, 6-bisphosphatase and NADP, malate dehydrogenase have provided valuable information about the mechanisms involved. This signal of the regulatory protein thioredoxin is transmitted to specific target enzymes, resulting in their activation in some cases, such as fructose 1, 6-bisphosphatase. The thioredoxin-linked activation is enhanced by an effector, e.g., fructose 1, 6-bisphosphate substrate. Inactive target enzymes observed upon darkening appears to take place by reversal of the reduction activation pathway. Oxygen converts the thioredoxin and target enzyme from the reduced state HHS to the oxidized state S, and, in so doing, leads to inactivation of the enzyme. The last are regulated directly by thioredoxin. The first, rubisco, is regulated indirectly by a thioredoxin accessory enzyme, rubisco activase. C2 cycle, photorespiration, respiration associated with photosynthetic tissues. It was discovered by Decker and Teo in 1959. Photorespiration, light-dependent utilization of oxygen and release of carbon dioxide by the photosynthetic organs of a plant. Normally photosynthetic organs do the reverse in the light, i.e., uptake of CO2 and release of O2. Therefore, photorespiration is difficult to demonstrate. It is inferred from ITs in the rate of net photosynthesis when oxygen concentration is increased from 2-3% to 21%, sudden increased evolution of O2 when an illuminated green organ is transferred to dark. The site for photorespiration is chloroplast. Peroxisome is required for completing the process. RUBP carboxylase is changed to RUBP oxygenase. This happens at high temperature and high oxygen concentration. At high temperature and high oxygen concentration, the affinity of RUBP carboxylase for Ca2 decreases and the affinity for O2 increases. High temperature occurs in tropical areas. Therefore, tropical plants are the major sufferers. Temperature, RUBP carboxylase functions as oxygenase and instead of fixing carbon dioxide, oxidizes ribulose 1,5-biphosphate to produce phosphoglyceric acid and phosphoglycolate. Phosphoglycolate is hydrolyzed to form glycolate. Glycolate usually passes into peroxisome of a mesophyll cell and forms glyoxylate. Glyoxylate is aminated and gives rise to amino acid glycine. Inside mitochondrion and even cytoplasm, the 2 molecules of glycine condense to form a molecule of serine. CO2 and ammonia are released in the process. Serine can further be deaminated to form PGA. The latter passes into chloroplasts for synthesis of photosynthetic products as well as photorespiration. Since photorespiration involves the synthesis of 2 carbon compounds, it is also called C2 cycle. Photorespiration does not produce energy or reduce power. Rather, it consumes energy. Further, it undoes the work of photosynthesis. It may reduce photosynthesis up to 50%. Therefore, photorespiration is a highly wasteful process. This happens only in case of C3 plants. C4 overcome the problem of photorespiration by performing Calvin cycle in the interior of leaves, bundle sheath cells, where both temperature and oxygen are lower. They have further ensured high CO2 supply to cells performing Calvin cycle. Institute for Advanced Studies, Jodhpur 10. Institute for Advanced Studies, Jodhpur 11. The biological function of photorespiration is unknown. Although the C2 oxidative carbon cycle recovers 75% of the carbon originally lost from the Calvin cycle as 2-phosphoglycolate, why does 2-phosphoglycolate form at all? One, that the formation of 2-phosphoglycolate is a consequence of the chemistry of the carboxylation reaction, which requires an intermediate that can react with both CO2 and O2. Such would have had little consequence in early evolutionary times if the ratio of CO2 to O2 in air were higher than it is today. However, the low CO2, O2 ratios prevalent in modern times are conducive to photorespiration, with no other function in the recovery of some of the carbon present in 2-phosphoglycolate. Another possible respiration is important, especially under conditions of high light intensity and low intercellular CO2 concentration, e.g., when stomata are closed because of water stress, to dissipate excess ATP and reducing power from light reactions, thus preventing damage to the photosynthetic apparatus. Arabidopsis mutants that are unable to photorespire grow normally under 2% Ca2, but they die rapidly if transferred to normal air. Residents from work with transgenic plants that photorespiration protects C3 plants from photooxidation and photoinhibition. Further work is needed to improve our understanding of the function of photorespiration. C4-DICARBOXYLIC acid pathway, Hatch-Slack pathway, C4 pathway. It was worked out by Hatch and Slack 1965-1967. 1965 found that labeled carbon dioxide L4C02 assimilated by sugarcane leaves first appeared in a 4-carbon compound oxaloacetic acid OAA or oxaloacetate. This found at a regular mode of CO2 fixation in a number of tropical plants, both monocots and dicots, e.g., maize, sugarcane, sorghum, panicum, pennisetum, atriplex, amaranthus, salsola, etc. These plants are called C4 plants because of the first stable photosynthetic product being a 4-carbon compound. Other plants are C3 plants. C4 plants often live hot, arid and saline habitat. They have Kranz anatomy. In Kranz, the mesophyll is indifferentiated and its cells occur in concentric layers around vascular bundles having large bundle sheath cells. The mesophyll and bundle sheath cells are connected by plasmodesmata or cytoplasmic bridges. The chloroplasts of the mesophyll cells are smaller. They have well-developed grana and a peripheral reticulum but no starch. The chloroplasts of the bundle sheath cells are larger. They have ill-defined grana, a peripheral reticulum and starch grains. Institute for Advanced Studies, Jodhpur 12. In C4 plants, initial fixation of carbon dioxide occurs in mesophyll cells. The primary acceptor of C02 is phosphoenol pyruvate or PEP. combines with carbon dioxide in the presence of PEP carboxylate PEPCO to form oxaloacetic acid or oxaloacetate. PEP carboxylase PEP plus CO2 plus H2O oxaloacetic acid plus H3PO4. Oxaloacetic acid is reduced to malic acid or aminated to form aspartic acid. Dehydrogenase. Oxaloacetic acid plus NADPH malic acid plus NADP plus. Dehydrogenase oxaloacetic acid plus NH3 plus NADPH aspartic acid plus NADP plus H2O. Malic acid or aspartic acid is translocated to bundle sheath cells through plasmodesmata. Inside the bundle sheath cells they are decarboxylated and deaminated in case of aspartic acid to form pyruvate and CO2. Malic enzyme. Malic acid plus NADP plus pyruvate plus CO2 plus NADPH. CO2 is again fixed inside the bundle sheath cells through Calvin cycle. RUBP of Calvin cycle is called secondary or final acceptor of CO2 in C4 plants. Pyruvate is sent back to mesophyll cells. Here, it is changed to phosphoenol pyruvate. Energy is required for this. The same is provided by ATP. The latter is changed into AMP, adenosine monophosphate. Phosphobaconase pyruvate plus ATP PEP plus AMP plus H3PO4. Conversion of AMP to ATP requires double the energy than conversion of ADP to ATP. Therefore, actual requirement of energy is equal to 2 molecules of ATP. This energy is in addition to 3 ATP required for fixation of 1 molecule of CO2 through Calvin cycle. Therefore, C4 plants consume 5 ATP molecules per molecule of CO2 fixed instead of 3 ATP molecules for C3 plants. For the formation of a glucose molecule, C4 plants require 30 ATP while C3 plants utilize only 18 ATP. RUBP plus 6 CO2 plus 30 ATP plus 12 NAPDH 6 PEP plus 6 RUBP plus C6H12O6 plus 30 ADP plus 30 H3P04 plus 12 NADP plus. Institute for Advanced Studies, Jodhpur 13. Importance. 1. The C4 plants are considered to possess greater photosynthetic efficiency, for they can utilize CO2 until a level of 5 ppm is reached, but the Calvin cycle plants cannot utilize CO2 if the level falls below 40 to 50 ppm. 2. C4 plants can utilize greater light intensities and their temperature optima for photosynthesis exceeds those of C3 plants. 3. The chloroplasts of these plants seem to generate more ATP, which of course makes for improved cellular work. 4. The presence of extra peripheral reticulum in the chloroplasts of these plants indirectly suggests quicker transport of products and therefore greater utilization of light and CO2. CRASSULACEAN acid metabolism. This pathway, work by Ranson and Thomas 1960, and Ruham, Vines and Black 1973, is found in succulents, mostly members of Crassulaceae, Bryophyllum and Sedum, and a few members of Bromeliaceae, such as pineapple. It is a device designed to meet the pressures of heavy transpiration, arising from their xerophytic environment. Plants obtain their CO2 requirements during the night time when they keep their stomata open and as CO2 buildup occurs, the cell sap turns acidic. This process is called as dark acidification. In the fall time, the stomata remain closed, minimizing transpirational losses, but with the advent of light, the CO2 absorbed during the preceding night is utilized for photosynthetic purposes. The process of light deacidification then occurs. Thus, there is a time lag between absorption and reduction of CO2. This arrangement helps to lessen transpirational stress but is responsible for the extremely slow growth of these plants. Below is given a short account of this pathway. Institute for Advanced Studies, Jodhpur 14. Phase 1, dark acidification. In this phase, the reserve starch is broken to phosphenolpyruvate PEP through a number of respiratory reactions. PEP accept, the atmospheric C02 absorbed by the plant and produces malic acid. This happens in darkness when the stomata are open. Starch PEP plus NADPH2 plus CO2 malic acid plus NADP. Phase 2, light deacidification. During this phase, which occurs in light when the stomata are closed, malic acid is decarboxylated into pyruvic acid and CO2. The CO2 released is routed through Calvin cycle to synthesize a hexose. The pyruvic acid is used to build up starch whose stocks are depleted earlier. Malic acid pyruvic acid plus NADPH2 plus CO2 in plant. Calvin cycle events. C2 plus 12 NADPH2 plus 18 ATP plus 11 H2O fructose minus 6 phosphate plus 12 NADP plus 18 ADP plus 17 pi. The assimilatory power needed for Calvin cycle is provided by the events of light deacidification. Principle OR law of limiting factors. Optimum value of a factor is never constant. It depends upon the magnitude of other factors. We may continue to increase the magnitude of one or more factors without influencing the rate of reaction. In such cases it is found that a factor called limiting factor is holding the balance. A limiting factor is defined as a factor which is deficient to such an extent that increase in its magnitude directly increases the rate of the process. The effect of limiting factors was studied by Black 1 and Arm in 1905. He formulated the principle of limiting factors, which states that when a process is conditioned as to its rapidity by a number of separate factors, the rate of the process is limited by the pace of the slowest factor. In other words, the rate of a physiological process is limited at a given time by one and only one factor which is deficient. Factors influencing photosynthesis. External factors. 1. Carbon dioxide. CO2 concentration of the atmosphere is 0.03% or 300 ppm. It is a limiting factor as the available CO2 concentration is lower than the optimum for photosynthesis. Increase in its concentration up to 0.1% increases the rate of photosynthesis in most land plants. A decline is observed beyond it. When CO2 concentration is reduced, there comes a point at which illuminated plant parts stop absorbing carbon dioxide from their environment. It is known as CO2 compensation point or threshold value. At this value, CO2 fixed by photosynthesis is equal to CO2 evolved in respiration and photorespiration. The CO2 compensation point reflects the balance between photosynthesis and respiration as a function of CO2 concentration, and the light compensation point reflects that balance as a function of photon flux. 2. Light intensity. Plants are broadly classified into two groups depending upon their inability or ability to tolerate high light intensity. Shade plants, geophytes, e.g., oxalis, and sun plants, calliophys. At low light intensity, the rate of photosynthesis is reduced. There is a point in light intensity where there is no gaseous exchange in photosynthesis. It is called light compensation point. Light compensation point is defined as a point in light intensity when no gaseous exchange is observed between the environment and the photosynthetic organ illuminated by that light intensity. A plant cannot survive for long at compensation point because there is a net loss of organic matter due to respiration of non-green organs and respiration in dark. The light intensity at which a plant can achieve maximum amount of photosynthesis is called saturation point. It has 800-1000 feet candles, 10% of full sunlight in shade plants, 50-70% of full sunlight in C3 sun plants and up to 200% of full sunlight in C4 sun plants, e.g., sugarcane. Beyond saturation point, it is seldom realized in nature in C4 sun plants, the rate of photosynthesis begins to decline. The phenomenon is called solarization. I. Photoinhibition due to reduction in hydration and closure of stomata. E. Photooxidation or oxidation of photosynthetic pigments, intermediates and enzymes. 3. Quality of light. Maximum photosynthesis occurs in blue, violet and red regions of the light spectrum where most of the absorption is carried out by chlorophylls. Red light favors carbohydrate accumulation while blue light stimulates protein synthesis. Minimum photosynthesis occurs in the green wavelengths. Plants growing under the canopy of other trees receive very little red and blue violet light because of its absorption by leaves of the canopy. They receive more of green light that is transmitted through the tree. Leaves. As a result, the rate of photosynthesis of herbs, shrubs and OT, for or undergrowths in a forest is comparatively low. Ultraviolet rays are harmful. Institute for Advanced Studies, Jodhpur 15. 4. Duration. Continuous photosynthesis can occur in continuous illumination without any harm to the plant bonding. 1949, though the rate of photosynthesis may slightly decline after 6 days. 5. Temperature. It does not influence light reactions of photosynthesis but affects the enzyme-controlled dark reactions. At which most plants start photosynthesis is 0 degrees minus 5 degrees Celsius, but it can be as low as 20 degrees Celsius for lichens and minus 35 degrees Celsius for some gymnosperms. The maximum temperature at which photosynthesis can occur is 55 degrees Celsius in some desert plants and 75 degrees Celsius for hot spring algae. The optimum temperature is 10 degrees minus 25 degrees Celsius for C3 plants and 30 degrees 45 degrees Celsius for C4 plants. When temperature is increased from minimum to optimum, the rate of photosynthesis doubles for every L0 OC rise in temperature. Above the optimum temperature, the rate of photosynthesis shows an initial increase for short duration that later declines. This decline with time is called time factor. 6. Oxygen. Small quantity of oxygen is essential for photosynthesis except in some anaerobic bacteria. C3 plants show optimum photosynthesis at low oxygen concentration. Example, Buman et al. 1968, found in beans twice the rate of photosynthesis at 2.5% oxygen as compared to normal atmospheric concentration. Possible reasons are I. Oxygen takes part in oxidation of photosynthetic pigments, intermediates and enzymes in the presence of strong light photooxidation. E. Oxygen is a strong quencher of excited state of chlorophyll. E. Oxygen competes with C02 for reducing power. However, this effect is not known in C4 plants. IV. It converts RUBP carboxylase to RUBP oxygenase. At a very high oxygen content, the rate of photosynthesis begins to decline in all plants. The phenomenon is called Warburg effect. 7. Water. Water supplies H+ and electrons for carbon dioxide fixation. However, less than 1 degree of the total water absorbed is utilized in photosynthesis. The rest is lost in transpiration. Even a slight transpiration reduces the leaf hydration that cuts down photosynthesis by causing stomatal closure and hence decreased CO2 absorption, loss of leaf turgidity, reduced absorption of solar radiations and decreased enzymatic activity. Thus photosynthesis is more sensitive to dehydration than any other metabolic process. 8. Air pollutants. Dust and smoke particles present in the atmosphere reduce photosynthesis by reducing light penetration and forming a layer over the plants. Sulfur dioxide, nitrogen oxides, hydrogen fluorides and other air pollutants also decrease photosynthesis. 9. Minerals. Magnesium is a component of chlorophyll. Fe, Cu and Mn are required for synthesis of chlorophyll. Mn and Cl are linked to photolysis of water. P as phosphate is essential for ATP synthesis. Enzyme activators of photosynthesis include potassium and sulfur. Lower availability of any of these minerals reduces rate of photosynthesis. Internal or plant factors. 1. Age. As a leaf develops, the rate of photosynthesis rises with the age till it becomes maximum at full maturity. Afterwards, the rate of photosynthesis begins to decline. Institute for Advanced Studies, Jodhpur 16. Respiration, citric acid cycle, plant mitochondrial electron transport and ATP synthesis, alternate oxidase. Respiration, the oxidative breakdown of organic compounds. 1. The overall process in respiratory substrates. Earlier in this chapter it was discussed how respiration counterbalances photosynthesis in the biosphere, oxidizing the C fixed in photosynthesis to CO2 and water again. Most of the respiration is aerobic and utilizes the O2 produced in photosynthesis, so that the O2 is recycled as well. The primary function of respiration is to provide for the energy needs of living cells. Some of the potential energy of the oxidizable substrates is conserved in ATP. Respiration is sometimes described as being the reverse of photosynthesis. Chemically, the overall result of respiration is the reverse of that of photosynthesis. Taking carbohydrate as the end product of photosynthesis or the substrate of respiration, one can write. To the reaction mechanism, however, respiration as a whole is not the exact reverse of photosynthesis, although there are many common intermediates, and some reactions do run in the reverse direction in the two processes. Quantitatively, the amount of photosynthesis by a plant must exceed its respiration or a positive mass balance would be impossible. It has been estimated that a flowering plant respires daily 30 to 70% of its photosynthesis, not counting any photorespiratory losses that may have occurred. In most plant tissues, carbohydrate is the main respiratory substrate, entering the oxidative pathways as hexose sugars. Such sugars are metabolically reactive and are not stored in cells in large amounts. Carbohydrate is stored as polysaccharides of which starch, a glucose polymer, is the most common. It is insoluble in formed starch grains in plastids. Some species store fructosins, soluble polymers of fructose, in vacuoles. Disaccharide sucrose is the main vacuolar store in yet other plants, and sucrose is also the most common form in which carbohydrate is transported in plants. All these more complex carbohydrates have to be hydrolyzed to their hexose monomers for respiration. Many seeds store lipids as oil, and this can serve as respiratory substrate, although most of the lipid store is converted to carbohydrate before being respired. Protein is not commonly utilized as a respiratory substrate, but sea skeletons from amino acids can enter the respiratory pathways. During starvation, or senescence, and during reserve mobilization in seed storage tissues, there is large-scale hydrolysis of proteins and respiration of at least part of the amino acid pool. Much of the basic biochemistry of respiration is common to organisms from all the living kingdoms, though some details may be specific to plants. With reference to photosynthetic organisms, the respiration which passes through the universal respiratory pathways, as described below, is often termed dark respiration, to distinguish it from the unique, photosynthesis-linked photorespiration. The so-called dark respiration still proceeds in photosynthetic tissues in the light. As in the case of photosynthesis, which proceeds in two stages, one can distinguish two stages in respiration: the stage of breakdown of substrate, which yields CO2 and reduced coenzymes, NADPH, and the stage of terminal oxidation, which achieves oxidation of the reduced coenzymes. Both of these stages yield ATP, but by far the greater proportion is produced during terminal oxidation. Two pathways of substrate breakdown: glycolysis. The word glycolysis means sugar breakdown. It brings about the oxidative breakdown of glucose to pyruvate, as illustrated in Figure 1. First converted to fructose 1,6-bisphosphate by phosphorylation at the expense of ATP, catalyzed by hexokinase, followed by isomerization and a second phosphorylation by phosphofructokinase. Fructose 1,6-bisphosphate is cleaved by aldolase to the two tri-sugar phosphates, 3-phosphoglyceraldehyde and dihydroxyacetone 3-phosphate. Then comes the oxidative reaction. The 3-phosphoglyceraldehyde is oxidized by glycerol dihydrophosphate dehydrogenase to 1,3-phosphoglycerate PGA, and the coenzyme NAD+ is reduced. The 1,3-PGA donates the phosphate group to ADP to synthesize ATP, catalyzed by PGA kinase, a process known as substrate-level phosphorylation. Molecular rearrangements: phosphoenol pyruvate, PEP, is formed in pyruvate kinase catalysis, a second substrate-level phosphorylation to give the end product of glycolysis, pyruvate. Seconds from the oxidation step onwards proceed twice per molecule of glucose. 4 ATP per 1 glucose can be formed, but 2 ATP are used to prime the system, so that the net gain is 2 ATP per 1 glucose. C6H12O6 plus 2 ATP plus 2 Namibian dollars plus C3H4O3 plus 4 ATP plus 2 NADH2. Although still in organic combination, INO-CO2 has been evolved and most of the potential energy of the glucose is still present in the pyruvate and the NADH. The net gain of 2 ATP represents a very small percentage of the potential total energy. It may also be noted that no O2 has been used. Glycolysis is an anaerobic pathway. Substrates can enter the glycolytic pathway at several points. Fructose 6-phosphate may come from the hydrolysis of fructans or sucrose, or from the PPP. Triose sugar phosphates are transported out of chloroplasts in the light and may also be derived from the PPP. Starch hydrolysis by the starch phosphorylase enzyme produces glucose 1-phosphate, which is easily isomerized to glucose 6-phosphate. When such phosphorylated sugars enter glycolysis, one or both of the priming reactions with ATP is bypassed and the ATP gain is correspondingly greater. Intermediate drawn into other metabolic sequences from the glycolytic pathway at various points before pyruvate is produced. The complete glycolytic sequence is located in the cytosol. In plant cells, however, isozymes of all the glycolytic enzymes are additionally found in plastids. Isozymes are events of an enzyme, catalyzing the same reaction, but differing slightly in structure and properties such as substrate affinity. Isozymes may be coded by different genes, or their differences may result from post-transcriptional or post-translational modifications. The function of glycolysis in plastids appears to be the production of pyruvate for fatty. Institute for Advanced Studies, Jodhpur 17. Acid biosynthesis, a process confined to plastids and plant cells and particularly active in seed tissues synthesizing oil as nutrient store. Photosynthetic CO2 metabolism involves several reactions catalyzed by glycolytic isozymes, but in the opposite direction to respiratory reactions. For instance, in the C3-cycle chloroplast aldolase catalysis, the condensation of triose phosphates to fructose 1,6-bisphosphate. In glycolysis, cytosolic aldolase cleaves the sugar. Institute for Advanced Studies, Jodhpur 18. The pentose phosphate pathway, PPP. This reaction series is also known as the hexose monophosphate shunt, or as the oxidative pentose pathway, OPP, to distinguish it from an alternative name for the C3 cycle, which is sometimes termed the reductive pentose phosphate pathway. The PPP is located in the cytosol. It is outlined in Figure 2. The starting substrate for the PPP is glucose 6-phosphate, or glucose followed by the hexokinase reaction. The commences with the oxidation of glucose 6-phosphate to 6-phosphoglucanate, followed by an oxidative decarboxylation of the glucanate to ribulose 5-phosphate with the release of CO2. The enzymes for these two reactions are glucose 6-phosphate dehydrogenase and phosphoglucanate dehydrogenase. The coenzyme which receives the H equivalents from both reactions is NADP+. The ribulose 5-phosphate is recycled to glucose 6-phosphate. 6-ribulose 5-phosphate 5-glucose 6-phosphate. The reactions here are largely a reversal of the C3 cycle reactions which regenerate the CO2 except for ribulose 1,5-bisphosphate, but no ATP is expended in the chloroplast and the cytosol each has a distinctive set of isozymes, just as for glycolytic ones. The balance sheet for the PPP is shown in equations. To be able to work with whole molecules, one must start with 6 hexose C6 molecules. Phosphates have been omitted for simplicity. This reduces to. The PPP thus does achieve the complete oxidation of glucose to CO2, but without ATP formation, there is no substrate-level phosphorylation involved. The mission of the PPP is thought to be the provision of NADPH for reductive biosynthesis, e.g. lipid formation, and for the production of metabolic intermediates. The pentose sugars can be utilized for nucleotide synthesis. Some of the NADPH may be oxidized by mitochondria with ATP formation. The oxidative pentose phosphate pathway plays several roles in plant metabolism. Product of the two oxidative steps is NADPH, and this NADPH is thought to drive reductive steps associated with various biosynthetic reactions that occur in the cytosol. In plastids, such as amyloplasts, and in chloroplasts functioning in the dark, the pathway may also supply NADPH for biosynthetic reactions such as lipid biosynthesis and nitrogen assimilation. Because plants are able to oxidize cytosolic NADPH via an NADPH dehydrogenase localized on the external surface of the inner membrane, some of the reducing power generated by this pathway may contribute to cellular energy metabolism. That is, electrons from NADPH may end up reducing O2 and generating ATP. The pathway produces ribose 5-phosphate, a precursor of the ribose and deoxyribose needed in the synthesis of RNA and DNA, respectively. Another intermediate in this pathway, the 4-carbon erythrose 4-phosphate, combines with PEP in the initial reaction that produces plant phenolic compounds, including the aromatic amino acids and the precursors of lignin, flavonoids, and phytolexins. Early stages of greening, before leaf tissues become fully photoautotrophic, the oxidative pentose phosphate pathway is thought to be involved in generating Calvin cycle intermediates. Control of the oxidative pathway. The pentose phosphate pathway is controlled by the initial reaction of the pathway catalyzed by glucose 6-phosphate dehydrogenase, the activity of which is markedly inhibited by a high ratio of NADPH to NADP+. However, little operation of the oxidative pathway is likely to occur in the chloroplast because the end products of the pathway, fructose 6-phosphate and glyceraldehyde 3-phosphate, are being synthesized by the Calvin cycle. Thus, mass action will drive the non-oxidative interconversions of the pathway in the direction of pentose synthesis. Moreover, glucose 6-phosphate dehydrogenase will be inhibited during photosynthesis by the high ratio of NADPH to NADP+ in the chloroplast, as well as by reductive inactivation involving the ferredoxin thioredoxin system. Institute for Advanced Studies, Jodhpur 19. The Krebs cycle. The Krebs cycle is the reaction series which achieves the complete oxidation of pyruvate coming mainly from glycolysis to CO2. It is located entirely and exclusively in the mitochondria. The reactions are summarized in Figure 3. The pyruvate with 3 atoms first loses CO2 by oxidative decarboxylation catalyzed by the enzyme pyruvate dehydrogenase, which also links the remaining 2C fragment, an acetyl moiety, to coenzyme of producing acetyl-CoA. This condenses with the 4C acid oxaloacetate to form a 6C acid citrate. Then, as indicated in Figure 3, there follows a series of molecular rearrangements, oxidation steps and oxidative decarboxylation steps, until the equivalent of the pyruvate has been converted to CO2 and the oxaloacetate has been regenerated. There's one substrate-level phosphorylation producing ATP, associated with the oxidation of 2-oxoglutarate. At 3 steps, a molecule of water is added to the reactants. The overall final balance sheet for respiration shows water as a product, but water is also a substrate in respiration. Compare with photosynthesis, where water is not only consumed as indicated by the overall equation, but is also a product. 5 pairs of H equivalents removed from the substrates in the Krebs cycle reduce NAD+, except for the succinate oxidation step, where no coenzyme is involved. The enzymes are present in the mitochondrial matrix, but succinate dehydrogenase is again an exception, being bound to the crystal membrane of the mitochondrion. Between them the Krebs cycle and glycolysis can carry out the complete oxidation of glucose. With 2 H representing the pairs of H equivalents removed from substrates, one can write. Institute for Advanced Studies, Jodhpur 20. The H equivalents can be oxidized in the terminal oxidation reactions using molecular O2, and producing 12H2O per 1 molecule glucose. Like glycolysis in the PPP, the Krebs cycle can oxidize intermediates fed in at any place in the sequence. Its role in this respect is discussed later. The Krebs cycle is also an important source for metabolic intermediates. Malate, oxaloacetate and 2-oxoglutarate are C-skeletons for amino acids. Running down of the cycle by removal of intermediates is prevented by the occurrence of anaplerotic reactions which replenish it. 1. The carboxylation of PEP by PEP carboxylase to give oxaloacetate. This is of course the initial reaction in C4 and CAM photosynthesis, but it takes place also in non-photosynthetic cells, at a lower rate. Malate can be produced by malic enzyme from pyruvate, CO2 and NADPH. The citric acid cycle of plants has unique features. Citric acid cycle reactions outlined in Figure are not all identical with those carried out by animal mitochondria. For example, the step catalyzed by succinyl-CoA synthetase produces ATP in plants and GTP in animals. The plant citric acid cycle that is absent in many other organisms is the significant activity of NAD plus malic enzyme, which has been found in the matrix of all plant mitochondria analyzed to date. This enzyme catalyzes the oxidative decarboxylation of malate. Malate plus NAD plus pyruvate plus CO2 plus NADH. The presence of NAD plus malic enzyme enables plant mitochondria to operate alternative pathways for the metabolism of PEP derived from glycolysis. As already described, malate can be synthesized from PEP in the cytosol via the enzymes PEP carboxylase and malate dehydrogenase. Malate is then transported into the mitochondrial matrix, where NAD plus malic enzyme can oxidize it to pyruvate. This action makes possible the complete net oxidation of citric acid cycle intermediates such as malate or citrate. Alternatively, the malate produced via the PEP carboxylase can replace citric acid cycle intermediates used in biosynthesis. Reactions that can replenish intermediates in a metabolic cycle are known as anaplerotic. For example, export of 2-oxoglutarate for nitrogen assimilation in the chloroplast will cause a shortage of malate needed in the citrate synthase reaction. This malate can be replaced through the PEP carboxylase pathway. The presence of an alternative pathway for the oxidation of malate is consistent with the observation that many plants, in addition to those that carry out cresylation acid metabolism, store significant levels of malate in their central vacuole. Electron transport and ATP synthesis at the inner mitochondrial membrane. ATP is used by cells to drive living processes, and chemical energy conserved during the citric acid cycle in the form of NADH and FADH2, redox equivalents with high energy electrons, must be converted to ATP to perform useful work in the cell. This O2-dependent process, called oxidative phosphorylation, occurs in the inner mitochondrial membrane. Institute for Advanced Studies, Jodhpur 21. Although similar in all aerobic cells, the electron transport chain of plants and fungi contains multiple NADPH dehydrogenases and an alternative oxidase not found in mammalian mitochondria. The electron transport chain catalyzes the flow of electrons from NADH to O2. For each molecule of sucrose oxidized through glycolysis in the citric acid cycle pathways, 4 molecules of NADH are generated in the cytosol and 16 molecules of NADH plus 4 molecules of FADH2 associated with succinate dehydrogenase are generated in the mitochondrial matrix. Reduced compounds must be reoxidized or the entire respiratory process will come to a halt. The electron transport chain catalyzes an electron flow from NADH and FADH2 to oxygen, the final electron acceptor of the respiratory process. For the oxidation of NADH, the overall 2-electron transfer can be written as follows: NADH plus H plus plus 1/2 O2 NAD plus H2O. The electron transport chain of plants contains the same set of electron carriers found in mitochondria from other organisms. Individual electron transport proteins are organized into 4 multiprotein complexes identified by Roman numerals I through IV, all of which are localized in the inner mitochondrial membrane. Complex I NADH dehydrogenase. Electrons from NADH generated in the mitochondrial matrix during the citric acid cycle are oxidized by complex I and NADH dehydrogenase. Electron carriers in complex I include a tightly bound cofactor, flavin mononucleotide FMN, which is chemically similar to Fad, and several iron-sulfur centers. Complex I then transfers these electrons to ubiquinone. 4 protons are pumped from the matrix to the intermembrane space for every electron pair passing through the complex. Ubiquinone, a small lipid-soluble electron and proton carrier, is located within the inner membrane. It is not tightly associated with any protein, and it can diffuse within the hydrophobic core of the membrane bilayer. Complex II, succinate dehydrogenase. Oxygenate in the citric acid cycle is catalyzed by this complex, and the reducing equivalents are transferred via the FADH2 and a group of iron-sulfur proteins into the ubiquinone pool. This complex does not pump protons. Complex III, cytochrome BC1 complex. This oxidizes reduced ubiquinol, and transfers the electrons via an iron-sulfur center, 2b-type cytochromes B565 and B560, and a membrane-bound cytochrome C1 to cytochrome circa. 4 protons per electron pair are pumped by complex III. Cytochrome C is a small protein loosely attached to the outer surface of the inner membrane and serves as a mobile carrier to transfer electrons between complexes III and IV. Institute for Advanced Studies, Jodhpur 22. Complex IV, cytochrome C oxidase. This complex contains 2 copper centers, CuA and Cub, and cytochromes Na3. Complex IV is the terminal oxidase and brings about the 4-electron reduction of O2 to 2 molecules of H2O. 2 protons are pumped per lectin pair. Structurally and functionally, ubiquinone and the cytochrome BC1 complex are very similar to plastoquinone and the cytochrome B6F complex, respectively, in the photosynthetic electron transport chain. Some electron transport enzymes are unique to plant mitochondria. In addition to the set of electron carriers described in the previous section, plant mitochondria contain some components not found in mammalian mitochondria. Note that none of these additional enzymes pump protons and that energy conservation is therefore lower whenever they are used. NADPH dehydrogenases, both Ca2+ dependent, attached to the outer surface of the inner membrane facing the intermembrane space can oxidize cytosolic NADH and NADPH. Electrons from these external NADPH dehydrogenases, NDEX NADH and NDEX NADPH, enter the main electron transport chain at the level of the ubiquinone pool. Plant mitochondria have 2 pathways for oxidizing matrix NADH. One flow through complex I, described in the previous section, is sensitive to inhibition by several compounds, including rotenone and pyracidin. In addition, plant mitochondria have a rotenone-resistant dehydrogenase, ND and NADH, for the oxidation of NADH derived from citric acid cycle substrates. The role of this pathway may well be as a bypass being engaged when complex I is overloaded, such as under photorespiratory conditions. An NADPH dehydrogenase, ND and NADPH, is present on the matrix surface. Very little is known about this enzyme. Most, if not all, plants have an alternative respiratory pathway for the reduction of oxygen. This pathway is the so-called alternative oxidase that, unlike cytochrome C oxidase, is insensitive to inhibition by cyanide, azide, or carbon monoxide. ATP synthesis in the mitochondrion is coupled to electron transport. Oxidative phosphorylation, the transfer of electrons to oxygen via complexes at IV is coupled to the synthesis of ATP from ADP and pi via the ATP synthase complex V. The number of ATPs synthesized depends on the nature of the electron donor. In an experiment conducted with the use of isolated mitochondria, electrons derived from internal matrix NADH give ADP shock phase ratios, the number of ATPs synthesized for 2 electrons transferred to oxygen, of 2.4 to 2.7. And externally added NADH each give values in the range of 1.6 to 1.8, while ascorbate, which serves as an artificial electron donor to cytochrome C, gives values of 0.8 to 0.9. Results such as these for both plant and animal mitochondria have led to the general concept that there are 3 sites of energy conservation along the electron transport chain, at complexes I, III, and IV. Aerobic respiration yields about 60 molecules of ATP per molecule of sucrose. The complete oxidation of a sucrose molecule leads to the net formation of 8 molecules of ATP by substrate level phosphorylation, 4 during glycolysis and 4 in the citric acid cycle. 4 molecules of NADH in the cytosol. 16 molecules of NADH plus 4 molecules of FADH2 via succinate dehydrogenase in the mitochondrial matrix. On the basis of theoretical ADP shock phase values, a total of approximately 52 molecules of ATP will be generated for sucrose by oxidative phosphorylation. The result is a total of about 60 ATPs synthesized for sucrose stable. Using 50 mol-1 12 kcal-mol-1 as the actual free energy of formation of ATP in vivo, we find that about 3010 kilojoules mol-1, 720 kilocalories mol-1 of free energy is conserved in the form of ATP per mole of sucrose oxidized during aerobic respiration. This amount represents about 52% of the standard free energy available from the complete oxidation of sucrose, the rest is lost as heat. This is a vast improvement over the conversion of only 4% of the energy available in sucrose to ATP that is associated with fermentative metabolism. Plants have several mechanisms that lower the ATP yield. As we have seen, a complex machinery is required for a high efficiency of energy conservation and oxidative phosphorylation. So it is perhaps surprising that plant mitochondria have several functional proteins that reduce this efficiency. Probably plants are less limited by the energy supply, sunlight, than by other factors in the environment, e.g., access to nitrogen or phosphate. As a consequence, adaptational flexibility may be more important than energetic efficiency. In the following subsections we will discuss the role of the non-phosphorylating mechanisms and their possible usefulness in the life of the plant. The alternative oxidase. If cyanide 1M is added to actively respiring animal tissues, cytochrome C oxidase is inhibited and the respiration rate quickly drops to less than 1% of its initial level. But most plant tissues display a level of cyanide-resistant respiration that can represent 10 to 25%, and in some tissues up to 100%, of the uninhibited control rate. The enzyme responsible for this oxygen uptake has been identified as a cyanide-resistant oxidase component of the plant mitochondrial electron transport chain called the alternative oxidase. Electrons feed off the main electron transport chain into the alternative pathway at the level of the ubiquinone pool. The alternative oxidase, the only component of the alternative pathway, catalyzes a 4-electron reduction of oxygen to water and is specifically inhibited by several compounds, most notably salicyl hydroxamic acid (CHAM). When electrons pass to the alternative pathway from a ubiquinone pool, two sites of proton pumping at complexes 3 and IV are bypassed. Because there is no energy. Institute for Advanced Studies, Jodhpur 23. Conservation site in the alternative pathway between ubiquinone and oxygen. The free energy that would normally be conserved as ATP is lost as heat when electrons are shunted through the alternative pathway. How can a process as seemingly energetically wasteful as the alternative pathway contribute to plant metabolism? One of the functional usefulness of the alternative oxidase is its activity during floral development in certain members of the Araceae, the arum family, for example, the voodoo lily Pseuromatum vedatum. For pollination, tissues of the club-like inflorescence, called the appendix, which bears male and female flowers, exhibit a dramatic increase in the rate of respiration via the alternative pathway. As a result, the temperature of the upper appendix increases by as much as 25 degrees Celsius over the ambient temperature for a period of about 7 hours. During this extraordinary burst of heat production, certain amines, indoles, and terpenes are volatilized, and the plant therefore gives off a putrid odor that attracts insect pollinators. Acylic acid, a phenolic compound related to aspirin, has been identified as the chemical signal responsible for initiating this thermogenic event in the voodoo lily. In plants, however, both the respiratory rates and the rate of cyanide-resistant respiration are too low to generate sufficient heat to raise the temperature significantly, so what other roles does the alternative pathway play? It has been suggested that the alternative pathway can function as an energy overflow pathway, oxidizing respiratory substrates that accumulate in excess of those needed for growth, storage, or ATP synthesis. This view suggests that electrons flow through the alternative pathway only when the activity of the main pathway is saturated. Saturation is reached in the test tube in state 4. In vivo, saturation may occur if the respiration rate exceeds the cell's demand for ATP, i.e., if ADP levels are very low. However, it is now clear that the alternative oxidase can be active before the cytochrome pathway is saturated. The alternative oxidase makes it possible for the mitochondrion to adjust the relative rates of ATP production and synthesis of carbon skeletons for use in biosynthetic reactions. Another possible function of the alternative pathway is in the response of plants to a variety of stresses, phosphate deficiency, chilling, drought, osmotic stress, and so on, many of which can inhibit mitochondrial respiration. By draining electrons from the electron transport chain, the alternative pathway prevents a potential overreduction of the ubiquinone pool, which, if left unchecked, can lead to the generation of destructive reactive oxygen species such as superoxide anions and hydroxyl radicals. In this way, the alternative pathway may lessen the detrimental effects of stress on respiration. The uncoupling protein. Found in the inner membrane of mammalian mitochondria, the uncoupling protein can dramatically increase the proton permeability of the membrane and thus act as an uncoupler. As a result, less ATP and more heat is generated. Heat production appears to be one of the uncoupling protein's main functions in mammalian cells. It has long been thought that the alternative oxidase in plants and the uncoupling protein in mammals were simply two different means of achieving the same end. It was therefore surprising when a protein similar to the uncoupling protein was discovered in plant mitochondria. This protein is stress-induced and, like the alternative oxidase, may function to prevent overreduction of the electron transport chain. It remains unclear, however, why plant mitochondria require both mechanisms. The eternal, rotenone-insensitive NADH dehydrogenase, ND and NADH. This is one of the multiple NADPH dehydrogenases found in plant mitochondria. It has been suggested to work as a non-proton pumping bypass when complex I is overloaded. Complex I has a higher affinity for NADH, 10 times lower Km, than ND and NADH. NADH levels in the matrix, typically when ADP is available, state 3 complex I will dominate, whereas when at low levels in the matrix, typically when ADP is available, state 3 complex I will dominate, whereas when ADP is rate limiting, state 4 NADH levels will increase and ND and NADH will be more active. The physiological importance of this enzyme is, however, still unclear. Mitochondrial respiration is controlled by key metabolites. Of ATP synthesis, ADP and Pi, appear to be key regulators of the rates of glycolysis in the cytosol, as well as the citric acid cycle and oxidative phosphorylation in the mitochondria. Control points exist at all 3 stages of respiration. Here we will give just a brief overview of some major features. Institute for Advanced Studies, Jodhpur 24. Best characterized site of regulation of the citric acid cycle is at the pyruvate dehydrogenase complex, which is reversibly phosphorylated by a regulatory kinase and a phosphatase. Pyruvate dehydrogenase is inactive in the phosphorylated state, and the regulatory kinase is inhibited by pyruvate, allowing the enzyme to be active when substrate is available. In addition, several citric acid cycle enzymes, including pyruvate dehydrogenase and 2-oxoglutarate dehydrogenase, are directly inhibited by NADH. Lipid oxidation. Lipids are frequently stored in seeds as oil bodies, oleosomes, spherosomes, where they may account for over 50% of the tissue mass. The stored oils are triglycerides consisting of a glycerol molecule esterified with 3 long-chain fatty acids, mostly with 16 or 18 C atoms per chain. The first step in lipid oxidation is hydrolysis by lipase enzymes within the oil body to glycerol and fatty acids. Glycerol, a 3C sugar alcohol, is closely related to triose sugars and is converted to dihydroxyacetone 3-phosphate, which can then enter the glycolytic pathway. Fatty acids are activated by linkage to coenzyme A and are metabolized in the glyoxysomes, specialized microbodies of oily seeds, by 2 metabolic sequences. Firstly, the process of beta oxidation results in the cleavage of the fatty acid chain into 2C fragments, acetyl-CoA. Secondly, the glyoxysomes process the acetyl-CoA via the glyoxylate cycle to succinate in this scan, through a series of reactions in the cytosol and the mitochondria, finally be synthesized to sucrose. Most of the lipids stored in seeds is converted to sucrose and transported to the growing parts of the seedling. Some of the acetyl-CoA is utilized in the mitochondria of the storage cells as respiratory substrate in the Krebs cycle. 3 interactions of pathways. In the cell, none of the pathways of respiratory substrate oxidation functions in isolation. A summary overview of all the pathways, and their interrelationships, is presented in Figure 2.15. The first stage, hydrolysis, in Figure 4, does not involve oxidations and is not specific to respiration. The monomers produced from the polymer serve as substrates not only for respiration but for numerous other metabolic sequences. The free energy change associated with the hydrolytic reactions is minimal, less than 1% of the total available, and cannot support ATP synthesis. The incomplete oxidation stage produces a small number of fairly simple organic acids and the free energy content of the substrate falls by about 33%. Some of the energy is conserved in ATP during substrate level phosphorylations, and also in NADH. The organic gas feed into the complete oxidation stage, the Krebs cycle, which in association with the mitochondrial electron transport chain forms the final stage for most aerobic respiratory activity and where most of the ATP synthesis takes place. The PPP does not appear to fit the overall pattern in that it is a direct oxidative pathway. Sugars metabolized exclusively by the PPP do not pass through the Krebs cycle. However, the PPP and glycolysis share common intermediates, fructose 6-phosphate and triisphosphates. Since both reaction series take place in the cytosol, it is almost inevitable that some exchange of metabolites between the two pathways should occur. If, say, there is a high demand for NADPH, the flow of material through the PPP might be boosted by intermediates from the glycolytic sequence. A large demand for pyruvate, on the other hand, could result in the channeling of PPP intermediates into glycolysis. The PPP accordingly should be considered an integral part of a glycolysis, Krebs cycle, PPP network. This network in turn interacts with the rest of cellular metabolism via interchange of metabolites, at many steps. The provision of intermediates and reduced coenzymes is just as much a function of respiration as provision of ATP. Respiration, defined as a substrate breakdown process, is also the starting point of many biosyntheses. The ATP balance sheet and energy conversion efficiency of respiration. The theoretical balance sheet for respiratory ATP production is easily drawn up. If glucose is completely oxidized via glycolysis in the Krebs cycle, with terminal oxidation through cytochrome oxidase, one can add up the ATP molecules for one molecule glucose, table 2. The ADH from glycolysis, reacting with mitochondria from the outside, is oxidized with the production of only 2 ATP per NADH, whilst mitochondrially produced coenzyme oxidation yields 3 ATP per 1 NADH. The free energy of complete oxidation of glucose is 2880 kJ mol-1. The free energy of hydrolysis of ATP is highly dependent on factors such as ATP concentration and pH, but under cellular conditions is at least 42 kJ mol-1. Assuming a gain of 36 ATP per molecule of glucose is equivalent to an energy conservation of 36x42, 1512 kJ mol-1, or 52% of the total available, a very high degree of energy conservation. The value would be pushed even higher if phosphorylated sugars enter the glycolytic pathway. Calculation in table 2 is, however, based on an assumption of complete coupling of substrate breakdown and terminal oxidation to ATP synthesis. This is unlikely to be the situation in a cell, at least not in all circumstances. ADH, which is used in reductive reactions, does not yield ATP, and terminal oxidation by the alternative oxidase produces only 1 ATP per molecule of NADH oxidized. The 20H formed per molecule of glucose in the PPP could theoretically be oxidized by the mitochondria with the production of 24 ATP, but, as stated, the PPP probably provides NADPH mainly for reductive reactions rather than for terminal oxidation. It is therefore not possible to say exactly how much ATP is produced per glucose molecule in a particular situation. Anaerobic respiration. Occurrence and endurance of anaerobiosis in plants. Flowering plants are obligate aerobes. No flowering plant can complete its life cycle without O2. Most flowering plant organs, except dormant seeds, are killed by anoxia, complete lack of O2, within hours or a few days at most. Nevertheless, there are also many situations in which plant. Institute for Advanced Studies, Jodhpur 25. Tissues regularly survive at least hypoxia, O2 concentrations too low to support a normal level of aerobic respiration. Anaerobic respiration, fermentation, is therefore not unusual in plants. In hypoxia, both aerobic and anaerobic pathways operate simultaneously. Germinating seeds often undergo a period of O2 shortage during imbibition, because the testa can be highly impermeable towards O2. Also, before the cells are fully hydrated and expanded, there are hardly any air spaces between them. Respiration during this period has a high RQ, respiratory quotient, i.e. the ratio CO2 evolution, O2 uptake. For aerobic respiration, this ratio is unity. In anaerobic respiration, CO2 is evolved without any O2 uptake, raising the RQ. Analysis shows an accumulation of end products of fermentation, such as ethanol, in the seed tissues. When the emerging radical splits the testa, the RQ falls. Complete anoxia is, however, tolerated by seeds of very few species. Meristematic tissues carry out partly anaerobic respiration. Cells in meristematic regions are closely packed, without air spaces, making diffusion of O2 a problem. The vascular cambium lies quite deeply within plant organs, and outside it lies the phloem, a living tissue without intercellular air spaces and with a high demand for O2. In apical meristems, the mitochondria are immature, with few cristae per unit volume, so that their capacity for terminal oxidation is low. In both large fruits, tubers, thick stems and thick roots, the level